All right, it's time to start looking at photosynthesis. And we begin just by identifying what this is to make sure you know what it is. It's a leaf. That's pretty obvious. And we're going to start by describing the overall reaction that goes on through the process of photosynthesis. But this is overall, and it's important to understand that there are different, basically two different parts to photosynthesis, and we'll be looking at the details of those parts. But overall, what the process of photosynthesis does is it uses light energy to split water and carbon dioxide and rebond the elements and the energy into glucose. And oxygen is a byproduct. Okay? Overall, that's what photosynthesis does. And it's a magical process because it powers all life on the surface of the earth where there is light. All right, so if we zoom in here, and that's what these lines are representing, we zoom in on one plant cell. Hopefully you recognize that as a plant cell, because it has the three components that plant cells have that animal cells don't. Cell wall, central vacuole, and in this case the central vacuole is over top of the nucleus. That shadowy thing underneath the central vacuole is representing the nucleus, and the nucleolus within the nucleus. And then chloroplasts. And there are two chloroplasts in this diagram. And again, you must understand that these diagrams are caricatures, they're like cartoons, and the organelles are actually much more numerous and much smaller than what they show in the diagram. And then if we zoom in on a chloroplast, that's what this diagram is representing. The overall function of the chloroplast is photosynthesis, of course, so this process is what's taking place within each chloroplast, within each plant cell, within each leaf. The light, and this is where I was talking about, is divided into two parts: the light-dependent reactions and the light-independent reactions. The light-dependent reactions take place in the thylakoids. Notice that the membrane inside the chloroplast, which is known as the thylakoid membrane, is organized into discs, stacks of discs, and each disc is a thylakoid. Okay? So that's where the light-dependent reactions of photosynthesis take place, and that's where photosynthesis begins. The light-independent reactions then take place in the stroma. And the stroma you can think of as kind of like the cytoplasm of the chloroplast. It's the surrounding aqueous solution that fills the chloroplast and surrounds the thylakoid membrane. You'll notice that the page number here is 15A, and that's because I needed to insert a 15B so that we could take a look at plastids, because chloroplasts are a kind of plastid. Plastids are a whole family. Notice a family of organelles. Plastids are a whole family of organelles, double membrane bound organelles. If we've gone over chloroplast structure before, and if you'll recall, there's an inner membrane and an outer membrane, so that's what that means, double membrane bound. And they play a major role in plant metabolism, and they carry out various functions in plant metabolism, and they all have the same evolutionary origin. And that would be endosymbiosis 2. Remember, endosymbiosis is how explains the transition from prokaryotic to eukaryotic, and it happened twice. In photosynthetic organisms, it happened twice, and that's where plastids came from. Endosymbiosis 2 is the origin of plastids, including the chloroplast. One thing plastids will contain are pigments, like chlorophyll or other pigments that give the cell its color. So notice that in the diagram here, we're looking at a number of different kinds of chloroplasts that carry out a number of different functions. And this one makes and stores pigment. And there's chlorophyll, the pigment that we find in chloroplasts. So this organelle, or this plastid, is actually specialized to synthesize chlorophyll that's then going to be transferred to the chloroplast. So you'll notice the diagram kind of looks like a phylogenetic tree. In other words, it shows an ancestor, a common ancestor of all plastids, and that's this thing called a proplastid. And these various plastids then differentiate, just like cells differentiate into specialized cells. The organelles also have to differentiate into various functional organelles, and plastids are a great example of that. So in plants, plastids may differentiate into several specialized forms depending on the cell's function. So it depends on what the cell does as to what plastids it's going to have. Undifferentiated plastids, that's this thing called a proplastid, may develop into any of these variants or any of these kinds of plastids. So the proplastid gives rise to specialized plastids through the process of differentiation. Just like a stem cell will give rise to specialized cells through the process of differentiation. But here we're looking at organelles, not cells. So functions that that plastids can carry out are to store products and synthesize products and carry out biochemical pathways that are all regulated by enzymes. So storage, 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 synthesis, 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 enzymes control all the biochemical pathways to produce products through synthesis. And then, of course, chloroplasts are plastids, specialized plastid, that contains chlorophyll and can carry out photosynthesis. So chloroplast, photosynthesis. That's its main function. So that's the primary plastid that you need to know about. But you do need to know that chloroplasts are just one kind of plastid in a whole family of plastids that have the same evolutionary origin and the same developmental origin, from proplastids to all these different plastid types. So all plastids are prokaryote-like and contain their own DNA and reproduce through binary fission, which supports the whole concept of endosymbiosis, the whole theory of endosymbiosis. So again, endosymbiosis too is the theory as to where plastids came from in the first place. So this brings us back to that overall reaction of photosynthesis and who figured out that overall reaction and how it was discovered. Well, our story, the history here, starts with a scientist named von Helmont, and he carried out an experiment in the early 1600s that was basically the beginning of discovering how photosynthesis works. And what he did was, what he was trying to find out is, when a tree grows, and that's the experimental subject that he used, when a tree grows, where does all that matter come from? Because, matter is mass, has mass, and can be measured by weighing, and this tree is going to weigh a lot more than that tree, and that means that it gained a lot of mass. Where did all that mass come from? And it is, if you just observe it, it is kind of magical, because the tree just gets bigger and bigger. And how does it get bigger? Where does that material come from to make it get bigger? Well, von Helmont hypothesized that it came from the soil. So he designed an experiment, of course, to test this. And he grew a willow tree for 5 years, just one willow tree for 5 years. He did nothing but water it as it grew. At the beginning of the 5 years, he weighed both the tree and the soil separately. So when he planted the tree, he weighed the tree and he weighed the soil that he put in the pot. So he had weights for both the tree and the soil. And then he let it grow for 5 years, and then he weighed them again at the end. Very simple experiment. Took 5 years to perform it, but it's a very simple experiment. And so the independent variable in this experiment is time, which is kind of the ultimate independent variable, because, time's independent of everything. And the control group in this case is the tree at the beginning, the tree and the soil at the beginning. The experimental group would be the tree and the soil at the end of 5 years. Now remember that his hypothesis was that the matter for the growth of the tree came from the soil, right? That's why he weighed the soil. but his results showed that the tree increased greatly in mass, while the soil decreased very little. It did decrease a little bit, but not very much at all. So he concluded that the source of matter could only have been what he added throughout the entire 5 years, and the only thing he added was water. So he concluded that water is where the mass of the tree comes from, that trees are basically made out of the components of water, which you know is H2O. And he was partially right, but only partially. Water contributes only part of the carbohydrates that go into building the mass of the tree, because basically the whole tree is carbohydrate. There's proteins in there too, and lipids and nucleic acids, of course, but the large part of the mass of the tree is carbohydrate. So he discovered the hydrogen part of carbohydrate, the hydrate part of carbohydrate, as I like to say. So, of course, we need further experiments to try to figure out the rest of the process, because water is only part of it, and the hydro part of carbohydrate is only part of it, the hydrogen part of it. So Jan Sinneberg conducted an experiment in the mid-1700s, and he was trying to find out if carbon dioxide was required for plant growth. So he set up an experiment with 2 groups of plants under sealed bell jars. So these bell jars are just open at the bottom. So it's like an upside down jar. And you'll notice he put soda lime in the intake, the air intake in one of the jars, and he left the other jar open. So that's the independent variable here in the experiment. One jar is sealed, and any air that comes in has to go through soda lime, which removes the carbon dioxide, or it absorbs carbon dioxide. In the open one, carbon dioxide is free to come into the container. So the control group had no carbon dioxide. I'm sorry, the control group would be the one open to the air, and the experimental group would be the one that has no carbon dioxide. Because again, that's what he's trying to figure out, is if carbon dioxide is required. There was also sodium hydroxide solution inside the bell jar, and that also absorbs carbon dioxide. So he took 2 measures to make sure that there was no carbon dioxide within the bell jar for the experimental group. So as I just explained, the control group was open to the untreated air, including carbon dioxide, while the experimental group that contained sodium hydroxide and soda lime to absorb and remove the carbon dioxide. And that was the independent variable. So as you can see in the diagram, he watered both plants, but the ones without carbon dioxide did not grow. So the result is the experimental plants did not grow. The ones without carbon dioxide did not grow. And he concluded from that carbon dioxide is used by plants as a nutrient, and so it's required. And by the way, he surrounded the pot with a plastic bag, or not plastic, because plastic wasn't around at that time, but with a bag to prevent the gases from the soil, because there is bacteria in the soil that will produce carbon dioxide, and that could have given some carbon dioxide to the plant. So he took every measure to make sure that the experimental group had no carbon dioxide. So he was right, of course, but he only discovered part of it. Part of what makes a carbohydrate, he discovered the carbo part. So previous experiments discovered the hydrate part from water, and Sinneberg discovered the carbo part from carbon dioxide. And the last point here is that we now refer to that as carbon fixation. When inorganic carbon dioxide, because carbon dioxide, even though it's a carbon-containing compound, it's considered inorganic carbon. So when that's used and incorporated into organic compounds like glucose, like carbohydrates, that's what we refer to as carbon fixation. The inorganic carbon has been fixed into organic compounds. Around 1770 then, Joseph Priestley comes along and uses a very similar experimental setup to Sinneberg, but he was trying to find out if oxygen's involved. So he needed a way to remove the oxygen from inside the bell jar. But he used a bell jar, and you'll notice too that he has it upside down in a container of water. And that's just to seal it off so that atmospheric gas can't get in. And he burned a candle in the jar, and as you know, combustion, burning, requires oxygen, and so the candle eventually went out because all the oxygen was removed from inside the bell jar. And then he carefully inserted a plant into the jar, and after the plant was there for a while, he was able to relight the candle. And you can imagine that would be kind of tricky, because you'd have to get into the jar with something to light the candle with without getting it wet. But anyway, that was his experiment. So this just explains what I just explained here. So he used the bell jar to surround the plants. He burned a candle to remove the oxygen, placed the plant under the jar, and he was then, after a while, able to relight the candle. So, of course, the jars without the plants were the control group. Being able to relight the candle indicated the plants had released oxygen. So that's what he concluded. And of course he was right. We now know that plants release oxygen due to splitting of water at the beginning of photosynthesis. So you know again that water is H2O. The O is oxygen, and that's where the oxygen that plants release comes from. They split water at the very beginning of photosynthesis, and then the oxygen is released as oxygen gas. Since Priestley's experiment, a similar experiment has been done, but instead of burning a candle to use up oxygen, a mouse or a small animal can be placed under the bell jar with the plant. And so the mouse then is using oxygen, but remember it's also giving off carbon dioxide, which the plant needs. So the whole exchange here between the process of photosynthesis and the process of aerobic cellular respiration is represented within that bell jar. So that's the ecological link between the two processes. Remember, as I told you previously, the product of one are the reactants of the other. So the carbon dioxide that's produced by aerobic cellular respiration in the mouse was used by the plant, and then the plant releases oxygen, which is used by the mouse.